Good morning to all. On the behalf of Department of Pharmaceutical Medicine, Maharashtra University of Health Science, Nashik, I heartily welcome today's speaker and participant for online session on basics of preclinical safety evaluation. Today's speaker, Dr. Akshay Dwarkanath, currently serving as Senior Principal Scientist and Head, Regulatory and Computational Toxicology, Division of CSIR, Indian Institute of Toxicology, Lucknow, Uttar Pradesh. Dr. Dwarkanath having about 23 years experience in the area of preclinical safety evaluation and regulatory toxicology. Current responsibility include deputy test facility management, GLP test facility at CSIR and IITR. Sir also has played a key role in establishing state-of-the-art facilities meeting international regulatory requirements in the area of preclinical safety testing, both in private sector and government institutes. Prior to joining CSIR, Sir also served the pharmaceutical and drug discovery industry for about 12 years with Dr. Reddy's Limited, as well as Dabar Research Foundation and Adunius Therapeutic Limited. I request Dr. Dwarkanath sir to proceed today's session. Please, sir.
Yes, thank you, Dr. Vaibhav, and good morning to all those who are connected through the virtual medium. Hope I'm audible, clear?
Yes, sir. Yes, sir.
I will just share my screen and you can let me know if that is also seen.
Yes, sir. Please, sir.
Is my screen visible now?
Yes, sir. Yes, sir.
Fine. So I'll start.
Yes, sir.
I'll be speaking to you all for about 40-45 minutes, and then we'll have a short Q&A session. So basically, I'll just give an overview of what are the regulatory requirements for the conduct of preclinical safety evaluation. So a few slides on, say, the basics of toxicology and then regulatory toxicology, different areas in toxicology. Then I will talk to you a little bit about the OECD GLP requirements. And then I will touch upon a few of the toxicity studies, oral toxicity studies, which are usually required as a first stage of any preclinical safety evaluation program. So a little bit about toxicology. Of course, most of you, those who have joined this session, would already be aware of this. So as you all already know, initially, whenever we spoke about toxicology, it was mostly to do with the toxins, or which was mostly to do with the how the handling of the poisons. It had nothing to do with any of the drugs or chemicals. So mostly when we had issues of poisoning, so the poison would either be from a natural source or from a synthetic source. What exactly is the poison, how it is bringing about its effects, and based on that, what are the say antidotes. All this was initially included under the domain of toxicology. And as all of us know, though all of us who are got any connection with pharmacology, toxicology, and pharmaceutical sciences, this is a very, very famous, statement which all of us are aware of. It is everything can act as a toxin or can act as a poison. It is only the dose which actually decides. So, as I said, initially it was mainly to do only with toxins and poisons, but then over a period of time, people started realizing that even apparently harmless substances can also bring about toxic effects, or say certain harmful effects. And that's when the domain of toxicology was actually widened to include any adverse effects of any of the xenobiotics. So anything which is foreign to a biological system. It might be something which is consumed at certain levels which is safe, but then at certain higher levels the same thing becomes toxic. So toxicology then started including all other domains, like biomedical aspects, things which are used as biomedical devices, implants, which are basically used for say a therapeutic purpose, but if improperly used or if not handled properly, they can also result in certain amount of toxicity. Coupled with that was the various public health, environmental issues, air, water pollution. So when we talk about toxicity, or we talk about toxicology testing, institutes, and of course, like organizations are also involved in all these various aspects. Broadly, when we talk about any item which might act as a toxin, there are three major, say, categorizations which is done, under which it is studied. The first one is the mechanistic aspects of it. How exactly that particular item under study is going to bring about its harmful effect, toxic effect. So I might be using these words interchangeably: harmful, toxin, toxic, etc. So what are the changes that happen? How, at the cellular level or at the molecular level, what is the mechanism by which the particular the substance brings about its effect? Once you know the mechanism, we then need to actually describe it. How and what exactly is the effect? This comes under the realm of descriptive toxicology. So whether the effect is localized or is the effect generalized, whether it is a short-term effect or it is a long-term effect, whether it is a systemic effect, for example, affecting the entire cardiovascular system, or it is only restricted to a particular organ, that is the heart or the liver, whether it is an organ-specific toxic insult. All this is included under the realm of descriptive toxicology. And then finally, once we know the mechanism, we are able to actually describe what and how it happens. It is quite obvious that to be able to have a control, or to be able to manage a situation where you might be faced with an incidence of toxicity, we need to have certain rules and regulations. So rules and regulations actually come in at various points. So right from the time, if I take the example of a chemical which is being, say, which is being developed as a new drug, since most of you all here, the audience are mostly from the B Farm, M Farm background, I'm taking the example of a drug, of a new drug, say, discovery program. So there are regulations which come into play right from the stage of the preclinical testing, right from the stage of even before, say, efficacy testing of the particular chemical that you're developing. What are the kind of animal tests that are required before you actually even look at the first in human exposure? So you have certain regulations there. Once you clear that entire preclinical stage, you come into the clinical trials. There are an altogether separate set of regulations for your the clinical trials. Once clinical trials are also done, if it actually reaches a marketable stage, then there are certain rules, regulations to be fulfilled to allow the particular item to be marketed or not. And even after that, we do have certain regulations for post marketing surveillance. So as you can see, each and every stage, each and every step of a drug discovery program, there are certain regulations, there are certain guidelines which need to be fulfilled. And therefore it becomes very, very important to be able to actually uh, decide what are the kind of regulations, how much of regulations. So if you see the image on a screen, if you see on the left, if the regulatory toxicology say requirement is such that such a huge amount of data is generated, to actually go through that, to actually decide whether the particular chemical should be allowed further or no, would be a huge time taking task. So we need to actually strike a balance between what is it that we are trying to decide when we have certain regulation, what is it that we are trying to see. So we are mostly concerned with the human safety. So those which are required to ensure safe and human use should be included. But certain there might be certain other say regulations which might not always be required for all kinds or all types of drugs. So we should not follow the policy of one size fits all. Based on a case to case basis, the regulations are implemented. So that was just to very briefly about toxicology and how the connect between toxicology and actually regulatory. Now coming to preclinical safety and good laboratory practices. I am sure most of you would already be aware of what are GCP, GLP, GMP. So to put it in a very in the crisp manner, it is GXP. So where X can either be a clinical practice, it might be say a good laboratory practice, or it might be a good manufacturing practice. I'll be talking mostly about the good laboratory practices that need to be followed during in a preclinical safety evaluation scenario. So basically, GLP is a set of principles. This is just a textbook definition, a set of principles or a framework based on which preclinical safety evaluation studies are planned, performed, and then monitored. So you first plan a study, you write a study plan, you write a step-by-step procedure, then you actually perform the study based on your study plan, based on what you have mentioned, based on your materials, methods, etc. And then when the study is being undertaken, is being performed, it is also being monitored. This is where the quality assurance component of a GLP facility comes into play. So once you plan, perform, and you monitor the study, you have completed the study, and you have also recorded all your findings during the conduct of the study. You then prepare a report. And once the report is prepared, of course, it will be audited by the QAU. It will also be reviewed uh, by the study sponsor. So these are some terms which those of you who are into the GLP mode would know. a person or an individual or a organization who was actually carrying out the study would be the test facility, and the agency for whom the study is being conducted, that would be the sponsor. So a report is generated and is submitted to the sponsor, and at the same time you also have to archive the study. So GLP has OECD GLP guidelines has certain requirements that any GLP study has to be archived and has to be maintained in the archive for a minimum of 9 years from the time the study was conducted. Just one or two slides on how and when OECD GLP came into being. So initially, soon after the Second World War, the European countries and of course other Western nations, United States, etc., came together, but that was mostly for the economic or for the rebuilding of the businesses after the effects of the World War. So that was basically an economic cooperation that happened. Subsequently, there was also a certain amount of military cooperation between the countries, and that led to the formation of the NATO. And finally, 1960 onwards, once the military cooperation and the economic cooperation, etc., was achieved, then the countries started feeling the need for improved living standards to try and maintain sustainable development. And that's when OECD came into being for sharing of data. So when I say sharing of data, then we had this concept called mutual acceptance of data. Mutual acceptance of data, basically OECD member countries, they came to an understanding, or they came to a conclusion that all those nations who follow the OECD guidelines, OECD requirements, and if a particular study is conducted in a particular area, particular laboratory, that would be acceptable to all other member countries. I'll just go back one slide and show you. So OECD, when it came into being, there were initially 36 full-time member countries, which you can see on your screen. And over a period of time, about 56 other countries also joined in. So totally now there are around 92 or like 94 countries, of which there are 36 full-time members, and the remaining are observer countries. So under this concept of mutual acceptance of data, a study which is conducted in any of these 92 countries, the data generated from these studies would be acceptable to all the other countries, since they are all the signatories to the MAD agreement, mutual acceptance of data agreement. So under the mutual acceptance of data, there are two major requirements which all the member countries and observer countries need to follow. One are the OECD guidelines for the testing of chemicals, and next are the OECD principles of good laboratory practice. So OECD guidelines for testing of chemicals, under this, there are specific guidelines for different types of studies, for an acute study, for a chronic study. So I will be talking about that in the subsequent slides. And OECD principles of good laboratory practice, they basically touch upon, or they basically govern the way a GLP facility should be set up, the way it should be, so all the operational requirements, all the functional requirements of a GLP facility. Now, to tell you a little bit about what is the scope of the preclinical safety studies, where all are these preclinical safety studies say applicable. So preclinical, sometimes also referred to as a non-clinical, basically any study which is done before the phase wherein you go into humans, before your clinical trial phase, is called either a non-clinical safety or a preclinical safety. So this is applicable to different classes of chemicals or of test substance, pharmaceuticals, agrochemicals. Agrochemicals would include all your pesticides, weedicides, insecticides, etc. Then also your cosmetics, veterinary drugs, industrial chemicals, Of course, now for cosmetics, the animal studies are now absolutely banned. So for cosmetics, most of the preclinical testing is done using in vitro methods. And where all can these studies be conducted? They can be conducted in a GLP facility, in a laboratory, or some of these studies are also conducted as field studies. For example, ecotoxicology studies, when you need to study the effect on ecotoxicology models, fishes or say honeybees, earthworms. So these are all the studies which are conducted in field scenarios, field studies. The major objective of carrying out these studies are, and say the major objective of carrying out these studies under the GLP principles, under the GLP guidelines, basically to ensure uh, three major points with respect to the data, that is data quality, data repeatability, and data and traceability. As I said, the data which is generated in any of the GLP facilities is acceptable to all the other member countries, and hence it has to be of extremely high quality. You can ensure the quality of the data that you generate if you are very sure that you have followed all the all the guidelines for the conduct of that study. Again, taking the example of a preclinical study for a new chemical drug molecule, if you need to do a 28-day study, uh, you need to start from the very first step, that is uh, the animals that you're going to be using for your particular study. You have to be very sure about the good quality of the animals, because say a 28-day study with a drug being done in a laboratory in India, a 28-day study of the same drug being done in any European country should, and if they are following the same set of principles, same guidelines, the outcome should be like similar. If not identical, it should be similar. Then you come to data repeatability. Several times it does happen that a study needs to be repeated again at, say, after a few years, or just for certain amount of checks that have to be built in. Data repeatability you can ensure again by following the same set of principles, same guidelines, and also the data traceability. Once you conduct all the preclinical safety studies and you submit the data to a regulator, they will look at your data right from the first study that you conduct, which is the say the simple acute oral toxicity, till say the carcinogenicity study. This entire process itself goes into several years, 2 years, 5 years, maybe 7 years. So the the person, or rather the regulator, has to be able to trace back the conduct of the study, and hence these guidelines ensure the data traceability. Again, it helps in avoiding duplication. By the same say concept, a study conducted in India is acceptable in other geographies, and hence the study need not be duplicated there. Now I'll be talking to you about the different types of preclinical toxicity studies. Again, so since this is a workshop on the basics of preclinical, I'll be touching upon the very basic concepts in each of these, and anybody who is more interested in knowing any further details is free to contact either the organizers or me, and we can take it further. So now coming to the first set of studies are the in vitro studies. In vitro, as most of you would know, it is something which is done in a system either in glass or plastic, that is outside the biological system. It usually involves isolated tissues, organs, or cells. Nowadays, with like technological developments, we also have the 3D printed organs. So these are also being used for your initial screening studies, though not all of these models are have the regulatory acceptance, but that is actually a work in progress. So even for your in vitro say tests which were carried out which are carried out in cell lines and which have regulatory acceptance, a whole lot of preliminary studies were conducted until it actually obtained the regulatory acceptance. Similarly, the example of whole body or other 3D printed organ systems, which are now increasingly being used in toxicity testing, are undergoing various stages of validation for regulatory acceptance. In vitro has certain advantages, has certain disadvantages. It is quicker, you can, and it is less expensive as compared to animal studies. It can be used for a quick the screening. If you have, say, if you have 10 molecules that you want to take into animal studies, you can yeah, you can use an in vitro test system to screen all the 10 molecules, and out of 10 you might find only 2 which are really say promising. When I say promising, if you are developing it as an anti-diabetic, it so out of 10 there are only 2 molecules which actually show glucose lowering ability, so that you can obtain from your in vitro studies, and then you take it further into your in vivo. Of course, certain disadvantages also. Since it is in vitro, it is outside the biological system, it is not a true representation of the complete biological system. uh, any drug or chemical behaves very differently in a living system as compared to a in vitro system, and hence we need to be careful when we are selecting the type of in vitro test or type of in vitro study that we are planning to conduct. In vivo studies mostly done in various animal models, rodents, canine, non-human primates, ecotoxicology models, etc. This gives the advantage of seeing how the chemical will behave in a entire biological system or an entire animal body. This is an essential part of any of the preclinical safety testing, especially before we do the first in human dose calculation. There are again different types of in vivo testing which will come in the subsequent slides: acute toxicity, subchronic, chronic, multi-generational toxicology studies, reproductive toxicology studies, etc. And then we also have what are known as carcinogenicity studies, wherein for the animal model, if it is a rodent, the animal model is put through the say the particular test item is administered to the animal model for at least 60 to 70% of its entire lifespan, so which usually goes up till 2 years. In vivo, in vitro studies both should always be used say like complementary to each other. They cannot be used in isolation. So certain amount of data generation happens from your in vivo studies, certain amount of the data generation from in vitro studies, and both together the data is then used and analyzed to look at the further progress of the particular test item. One particular point of caution: data from in vitro studies should not be directly used to predict a biological response. As I said a couple of slides earlier, it is very obvious. In vitro will only give you an indication. It is kind of indicative. In vivo studies are more closely related to the actual biological response inside a biological system. Finally, the other kind of toxicology studies which is also being used are the in silico methods. In silico methods also are, so in silico methods are basically based on the huge amount of data that has already been generated for a particular class of compound. So if you have a new compound which has a particular structure, and you have data on other molecules which are closely related to the new molecule, either in structure or in composition, you can use your in silico methods for again screening. In silico methods are also used as a quicker screener than compared to the in vitro, and based on the data set that you already have or the data generated using an in silico method, you will be able to make a rough estimate of what are the probable say target organs, what are the probable effects that your new drug or chemical might bring about. It has one advantage of being a very high throughput mode of testing. It also helps in fulfilling the 3 hours requirement. Of course, as I keep saying again and again, there is nothing that can completely replace the in vivo method of preclinical safety, but then in silico, in vitro, both can be used to identify or can be used to shortlist from among a library of compounds that you have for safety evaluation. It can also help in certain gap filling of data. For example, if you have a particular a new drug or a chemical which you are developing as the same example of anti-diabetic, but you want to look at it on its you want to evaluate it for its cardiovascular effects, whether there are any side effects or no. So you can then run your in vitro or your in silico methods, and you will be able to find out if there are any other side effects which may or may not be highlighted in a in vivo study. There are various methods among the in silico. That is, you have the structure activity relationship data that you generate, and based on that, or you also have a read across evaluation. So when I say read across evaluation, based on data of related compounds, of similar class of compounds, of similar structure of compounds, you will look at the safety data that is already generated, and based on that you will come to a conclusion about the particular say chemical that is under study. Most of the guidelines that I will be talking about in the next few slides are pertaining to OECD guidelines. Apart from that, we have certain other guidelines, like for safety pharmacology studies, you have the ICH S7A, S7B. Then in India, we have the New Drugs and Clinical Trial Rules 2019. Then we have the Good Clinical Practice Guidelines, of course, which is not under the purview of the current talk. And then we have certain guidelines, certain requirements from the Central Insecticide Board, which mostly apply to the testing of all the agrochemicals. So coming back to OECD guidelines for the different types of studies, as you can see, most of your acute toxicity studies would be either say following the guideline 420, 423 or 425. In subsequent slides, you will know what are the subtle differences between these three different types of guidelines. All of them pertain to an acute toxicity study, but some small differences between them. Then you have the subacute, that is the 28-day repeat dose toxicity study, oral toxicity, which is done as per OECD 407. The 90-day oral toxicity study, which is done as per 408. 451, 452, 453, those are very closely related. 451 is specifically only for a carcinogenicity study. 453, which is not seen in our screen right now for obvious reasons, because it is very close to this. 453 mostly deals with the chronic toxicity study requirements, and 452 is a overlap between 451 and 453. So mostly 452 is a guideline which is preferred over 451 or 453. Coming to the acute toxicity guidelines, 420 or 423, the basic difference among these two is the number of animals that will be used and the number of doses that will be tested. As you can see, if you follow 420, you will need to start based on an initial sighting study which is conducted in one single animal. That means you may or may not have any prior information with respect to the particular chemical under test in terms of its proposed dosage, in terms of its efficacy dose, etc. 423 gives you the advantage that certain initial data, efficacy data, or maybe pharmacokinetic, pharmacodynamic data would be available. Based on that, you will say test either one of the four doses that you see on your screen: 550, 300 or 2000. Usually if prior data already indicates that it is a safe generally regarded as safe compound, then you start with the limit dose of 2000 milligram per kg. And if 2000 milligram per kg is safe, then the same dose is again repeated in another set of animals. So as you can see, totally 3 plus 3, 6 animals, your study will be completed. But if you follow 420, you will have to start with a sighting study and then slowly keep increasing the dose until you see certain amount of toxicity. So toxicity might either be in terms of just clinical science or toxicity in terms of mortality of the animal. And the number of animals used when you use 423 is 5 per group. In 423, it is only 3 per group. Certain commonalities between the guidelines are both are conducted in adult female rats or mice 8 to 12 weeks age, and the environmental conditions, acclimatization, etc. are all similar in both. It is a single oral dose that is given to the animals at the dose volume of 10 ml per kg. If it is an aqueous solution or suspension, you can go up till 20 ml per kg. They are observed for a period of for the first 4 to 6 hours. They are observed every 30 minutes, and then thereafter they will be observed once in a day. So when the observation includes changes in skin, fur, and then the mucous membrane, and any other say change in gait, or if there are any signs of any tremors, convulsions, etc. Important point is both the rats or mice, whichever is the species that is being used, have to be fasted prior to the dose administration. Since it is an acute study, there is just one single dose exposure for the animal. The animals need to be fasted before exposure to the test item. Body weights are measured on day 1, 7, and 14. So this is a 14-day observation study. You dose it on day 1, observe the animals for 2 weeks, 14 days, and on the 15th day, the animals are sacrificed. Once they are sacrificed, only a gross pathology observation is carried out. And if at all we see any signs in any of the organs, then we also collect organs uh, for histopathology. This is just one example of a flow chart. So this will be available in the OECD 423 guideline. You can download it from the OECD website. So if you start at 300 milligram per kg body weight, if you see one mortality, what is the route you follow? If you see 2 or more mortalities, what is the route that you follow? So this will be similar to if you start with 2000 mg per kg or 50 mg or 5 mg per kg. Next are the set of subacute and subchronic studies. So there is a lot of similarity between all these, except of course the duration. And as the duration of the study increases, the other parameters that you study also will increase. So OECD 407 is for a 4-week study. 408 is for a 90-day or a 180-day study, and chronic toxicity is 1 year or more. So in all these studies, we also have what is known as a recovery period. As I had said in one of my initial slides, so whether the effect is temporary or it is permanent, that we will be able to identify when we have a recovery period. So as long as the test item is being administered, we see certain toxic signs. And if you stop administering the test item, then there is a reversal of the toxic sign. That means it is a transient effect and it is not a permanent effect. The major endpoint or major I think there was a break in connection.
Yes, sir. Please continue.
I'll continue. Are my slides visible? Not yet. They're visible now?
Yes, sir, visible.
Okay, so I was talking about the subacute and subchronic and chronic toxicity studies. The number of animals in between, so the number of animals is actually different between the different studies, for obvious reasons, since the duration of the studies are different. All of them will have basically one control and 3 dose groups, low, mid, and high. And as I said in the previous slide, we will also have what is known as a recovery group. So OECD 407, that when you are doing a subacute study, 28-day study, you require only 5 animals per sex per group. But if you are doing a chronic toxicity study, then you need 20 animals per sex per group. Similarly, the size of your recovery group animal will either be 5 in a 28-day study or 10 in a longer duration study. Dose selection for these studies is based on your acute toxicity study and also on your PK/PD data. Environmental conditions, acclimatization, etc., are all uniform across the 3 studies, across the 3 types of studies. The time of dosing also it is usually preferred that the dosing is carried out in the morning between 9 to 11 in the morning, and also maintain the time of dosing across the entire duration of the study. General observations, mortality, morbidity, and then clinical signs, detailed clinical examination, which is done weekly, and a general observation, which is done on a daily basis. Body weight, feed consumption is also done. In a 28-day study, you do it once every 4 days, but in a longer duration study, you do it once in a week. Subsequently, you do once in 2 weeks. Certain amount of differences which are there between 407, 408, and 452. So now here you see only the subchronic and chronic studies. Functional observation battery, the grip strength, motor activity, urine analysis, ophthalmological examination, etc., are only a part of your longer duration studies. That is 90-day studies and more. So 90, 180, or even carcinogenicity studies. Urine analysis includes all the parameters. So both the physical and chemical parameters of urine. Now again, coming back to hematology, biochemistry, and the organ collection is again common among all the 3, 407, 408, and 452. These are all the parameters in the hematology, that is the complete blood picture. And under clinical chemistry, you have the liver function, kidney function, then you have your HDL cholesterol, LDL cholesterol, that is lipid profile. All this is part of your clinical chemistry evaluation in subacute, subchronic, or chronic toxicity studies. Organs which are collected are again the most of the target organs, that is the brain, heart, kidney, liver, the major organs. And subsequently, if you are expecting a particular effect, say for example, it is known to cause an effect, say on the muscles, then you also collect say tissue samples from the various muscles of your test system of the rat. Certain other organs are also collected for histopathology. So there are 2 different lists. The previous list is organs which are collected and also weighed. These are only collected for histopathology. These are a few of the test studies that you do in longer duration. That is on your left and right, both you see the grip strength evaluation. This is the system which is used for the motor activity, both the horizontal activity and the vertical activity. The distance that is traveled, the time between two motions, that is known as the resting time, horizontal, ambulatory, beam breaks, etc. are counted. Carcinogenicity study, apart from other chronic toxicity study. So the difference between a chronic toxicity and a carcinogenicity, you are also evaluating the kind of the cancerous growths that you might see. Well, how often? So you need to evaluate the incidence and what are the say the different organs in which these carcinogenic neoplasms are formed. In chronic toxicity study, it is mostly restricted to a target organ toxicity. But in terms of but connecting a carcinogenicity study, you are looking at the occurrence of the neoplasms. Certain amount of you also have to extrapolate between the carcinogenic effects based on the dose of the test item. So if it is a low dose exposure for a prolonged time versus high dose exposure for a shorter duration of time. So I will stop here, and we have maybe another 10-15 minutes. If at all anybody has any questions, I'd be happy to take. So as I said, I just attempted to give you all an overview of safety evaluation, preclinical safety evaluation, and any further specific topic of interest, please feel free to connect. Thank you.
Thank you, sir. Now I request participants, if you have any query or doubts, please ask. Any question from participants? Any question from participants? Okay, question है. Sir, question is from audience. Please explain acute toxicity OECD guideline again. Dr. Dwarkanath sir, can you hear me? The question is sir. Just wait a minute. Sir is connecting now. One question from audience.
I'm connected now. Please go ahead.
Yes, sir. One question from audience, sir. Acute toxicity OECD guideline.
Acute toxicity OECD has actually 3 guidelines. 423 is what is being used most common, because 423 has the advantages of a low number of animals while also giving you the output that you require. So for acute toxicity, you have 420, 423, 425. Which particular guideline you have to use would depend on what is the outcome that you're looking at from your acute tox. 423 is most commonly preferred.
Thank you, sir. Any other question, please?
So is this being live streamed on YouTube?
Yes, sir. Yes, sir.
So there would be some questions there if at all somebody has put on the chat
Yes.
box.
Okay, sir. Thank you.
Okay, thank you so much. In case anybody has any other questions, can get back to me.
Yes, sir. Yes.
Thank you
sir.
for the opportunity, Dr. Weber.
Yes, sir. Thank you, sir. Now, next speaker is Dr. Shailendra Singh. Is a principal scientist at Indian Institute of Toxicology, CSR, Lucknow. Sir, having more than 17 years of experience in pharmacokinetics and toxicokinetic studies. Prior to joining to CSIR, Dr. Singh served the Dr. Reddy's Lab, Hyderabad, then Biocon, Bristol, Mayor's Squibb R&D Center, Bangalore. Sir published more than 75 articles in various national and international journals. Also published several book chapters on having the 5 patents. Dr. Singh also received the prestigious awards like Young Mass Spectrometry Award by Indian Society of Mass Spectrometry, then Bristol Myers Squibb and R&D Star Award. Also member of National Scientific Committee like Bureau of Indian Standards, then Food Safety and Standard Authority of India. Also expert member of Joint FAO and WHO Expert Committee on Food Additives. I request Dr. Singh to proceed the session.
Thank you, sir, for the introduction. So I'm just sharing my slide now.
Yes, sir, please.
Okay, so good morning, everyone. So I will be talking about like how the toxicokinetics, along with I would include the pharmacokinetics, because these are like a terminology which are used interchangeably depending on the kind of study we are doing. And what is the role of these studies in pharmaceutical research? As Dr. Akshay Dwarkanath has already highlighted, some of the very important aspect, when we are talking about in terms of PK/PD correlation, and we are really trying to relate the toxicological effect with in terms of the doses, and we are selecting the doses for toxicological studies. There, the toxicokinetics understanding is very crucial for any kind of pharmaceutical molecule development. And even though other than the pharmaceutical, also if we talk about agrochemicals and other things, also this toxicokinetics understanding is very crucial for selecting the toxicological doses for pesticide toxicological studies. So as you can see that, and why this particular important is that understanding the kinetics, as you know that like all the substances are poison actually, if you see in true sense. It is like the dose which differentiates poison and remedy. Even if we take any drug molecule in more excess, like it can produce toxicity. So it is the dose, but how we have to select that dose, it depends on the two fundamentals parameter, which is toxicokinetics, or you can say the pharmacokinetics, and the pharmacodynamics, or you can say the word toxicodynamics. So I will use this terminology interchangeably. It is a similar in terms of principle, but only the effect which we are seeing makes them the different in terms of interpretation. Yeah. So in terms of definition, if you see, the toxicokinetics actually is like a study of the movement of the chemical around the body. It includes absorption. So those who are like from the, I think all the people are from the pharma background, so they understand this terminology very well, that the absorption primarily is like the transfer from the site of administration into the general circulation. So whatever things we take, any medicine by any route, it has to reach into the circulation. So that primarily governed by the absorption process. The distribution, which is like via the general circulation into the into and out of the tissues. As you know that compound once into the blood, it has to be distributed in the different tissues, and it has to come out from the tissue so that it can be eliminated from the body. So this is the term understanding of the ADME, particular the absorption, distribution, or biotransformation and excretion is actually makes this ADME terminology. And when we have the understanding of the toxicokinetics, how it will help us in regulatory framework is like it will help you to decide the dose and the species selection for toxicological studies. It helps you to the interpretation of the toxicity data, because several times we see that at certain doses we are seeing the toxicity, but when the doses are changing, toxicity are not seen. As we are increasing the doses, toxicity has not increased to the similar extent. So what could be the possible reason, like not seeing the effect as per the dose which we are increasing? So understanding of the relationship between the dose and the concentration inside the body actually helps you to interpret the toxicological data. Similarly, that another important area is the refinement of default uncertainty factor. This is like where the kinetics understanding is utilized, especially for the agrochemicals, where that presently that most of we cannot administer the agrochemicals in the human being. So most of the outcome, whatever has been decided, are based on the animal studies. So in order to define that doses for the agrochemicals, this toxicokinetics understanding is crucial, and it helps to define certain kind of uncertainty factor which in generally are taken into consideration when deciding the acceptable daily intake value for the human being. And it also plays very important role in interpretation of biomonitoring data. Let's say if in our day-to-day life we are exposed to certain chemicals, let's say from food, it could be like unintentional exposure to different kind of chemicals. It is starting from the, let's say, the toxins or the pesticide. But how to understand whether this exposure will cause any side effect or any adverse effect to ourselves or not? To understand those kind of things, the toxicokinetics again plays very important role. And biomonitoring study actually helps you to collect the biological sample randomly from the Indian population, and then we try to estimate the concentration of different kind of chemicals or their metabolites inside the blood and urine sample. And based on those concentration, we can correlate with the doses, those concentration, and we can understand whether we are going to have any harmful effect in terms of the random exposure to the chemicals through the environment. So in terms of like the definition, when you compare these both the terminology, toxicokinetics and the pharmacokinetics, the primary difference is the dose actually. If you see the TK is the primary purpose of the TK study is to correlate findings of toxicity, not therapeutic efficacy, with the corresponding level of exposure to an experimental drug or toxic compound. Dr. Akshay Dwarkanath also highlighted in his slide that interpretation of the data, where that how to correlate the exposure to the toxicity, where this toxicokinetics plays very crucial role. And the primary objective of toxicokinetics is to describe the systemic exposure achieved in the animals and its relationship to dose level and the time course of the toxicity study. So when we are studying the ADME of a molecule in the toxicological study, we call it the toxicokinetics. When we study the ADME properties of any given drug molecule, especially at the therapeutic level, we call it the pharmacokinetics. So fundamentally, both are same. We study absorption, distribution, metabolism, and excretion in both toxicokinetics and as well as in the pharmacokinetics. But how we are interpreting this data in terms of the toxicity and therapeutic efficacy makes this difference, toxicokinetics versus pharmacokinetics. So if you see that this is like slide is telling you that why we wanted to understand in terms of the, if you said take the drug molecule, how this pharmacokinetics data can be used, right? If you remember that in the Dr. Akshay Dwarkanath slide, he talked about that first in human dose selection, right? So prior to selecting any first in human dose, we have to carry out a lot of toxicokinetics or pharmacokinetic studies so that we can establish the correlation of the concentration versus effect and the toxic effect, the therapeutic effect as well as the toxic effect. So, and at the same time, those studies will help you to define the first in human dose. Let's say if animal studies tell us that this compound is toxic at certain doses, then definitely for human clinical trial, we can use some lower doses, because we now understand at what doses we are going to see the toxicity. So this whole understanding kinetics and dynamics will help you to decide the dose for the human being. As you can see, when we do the drug discovery, like there are thousands of molecules are synthesized for a particular class of molecule when we are trying to develop. Okay, but you cannot take all the molecules to the clinical studies, and these all molecule undergo like a lot of preclinical screening in terms of the kinetics properties of the molecule. So based on the pharmacokinetic property, we can identify the compound which has all the desirable property which is required for a drug molecule. So this will help you to really identify an ideal molecule during the preclinical phase, so that we can take the molecule which is most efficacious and not toxic, at the same time having the all ideal ADME properties, which is required for a drug to be a molecule, successful molecule. As you can see, this particular figure, it is showing you that time concentration phenomena. So as you know that the pharmacokinetic study will provide you the information on time versus concentration, whereas the dynamics will provide you the time versus response relationship. Because those who are doing the toxicological studies or any kind of efficacy studies in animal model or in vitro test system, what they most of the time they have information from animal studies, the time versus response relationship. If we combine the kinetics and dynamics together, then we can correlate the concentration with the response. So this is what is called the PK/PD. So by understanding the PK, by understanding the PD, we can correlate these, we can bring them together by using mathematical tool, and we can understand what is the relationship between the concentration and effect. So PK along with PD will tells the clinician how much, how often, and how long to dose a particular drug molecule. So this will help you to decide, as you can see on the right-hand side of this particular figure, where you can see that MEC is showing you the minimum effective concentration and MTC is showing you the maximum tolerated concentration. So from the therapeutic perspective, we wanted to have a molecule which remain within the therapeutic range till the time we are administering the next dose. Right. So by understanding the kinetic property of the molecule, we can decide that how much dose we have to administer, how often, because as you can see in this particular figure that after a particular time, the compound concentration is coming down and now the drug is become ineffective. So this tells us that now it is a time that we have to administer another dose, because now molecule is coming to the level which is not effective in the human being, because the concentration came down from which were responsive for the therapeutic effect. So in this way, we can decide that how often we have to administer and how long we have to administer depending on the disease condition. Another very important aspect is from the chemical point of view is that hazard versus risk. Actually, anything could be hazardous in the nature. Okay, in fact, even the water could be hazardous in nature if we are drinking too much. But the major important concept is the risk. Risk will we can understand the risk to any given chemical based on the understanding of its exposure. Let's say I know that the pesticides are toxic in nature; they are hazardous chemical. But if I am not exposed to them, then I am not worried. So exposure is actually become very important when we are trying to understand risk to any given chemical. So and again, that exposure you will only understand by having the toxicokinetics or pharmacokinetics understanding of a molecule. So you must have read in your maybe in your bachelor or even the masters that we talk about a lot about the linear kinetics versus non-linear kinetics, right? So this concept is actually tells you about that when the kinetics is linear, it means that we are expecting that as the dose we are changing, the systemic exposure, which is generally defined in terms of the area under the curve, we call it AUC. So when we changes the doses accordingly, that exposure is increasing. So this says that the doses as we are changing the dose, exposure is changing. This is called the linear kinetics. But they are the scenario where you will see the non-linear kinetics, where the exposure will not increase according to the dose. As you can see, these three figures which are given, which is showing you the linear correlationship, where as the dose is increasing, AUC is increasing. But where in the second one, you can see that exposure is increasing even the more than the dose increased. And in third one, you can see that the exposure is increasing less than the dose increase. So this particular scenario is very important from the toxicological study. Because as compared to the therapeutic, especially the therapeutic efficacy studies, there the doses are much less as compared to the toxicological study, where you will see that very high doses are given to the animals, because our aim is to find out any kind of toxicity of that molecule, so that we can establish what is the difference between the concentration which is toxic and the concentration which is within the therapeutic range. So this difference understanding is very important, and to understand that difference, very high doses are given to the animals. And because of the high doses, you will see that saturation of the kinetics of the molecule, and it becomes very difficult to increase the exposure sometimes in the animal studies. So understanding of the kinetics principle is become very important. And if we understand what could be the factors which might be contributing to this nonlinear kinetics, we can optimize the things accordingly, so that we can see that exposure within the linear range to interpret the toxicological outcomes. This is like some examples I have put in perspective that how that understanding of the exposure will help you to understand the toxicological outcome. let's say this first point says that subproportional increase of plasma exposure with dose might explain the unexpected absence in increase of severity with increasing dose. You may see that in most of the time what happened in the toxicology studies, when we increase the doses, we do not see the increase in the severity in the adverse response according to the dose increase. And the reason behind this could be very simple, because it is quite possible that when we are increasing the doses, we are not able to increase the exposure to the similar extent as the dose increase. And that is the reason you may not see the increase in the severity. Let's say if you do not have this data, then it becomes very difficult for you to interpret the toxicological finding. Similarly, if you see the second point, which says that marked pathological changes not present at low doses were observed after increasing the dose twofold. So suddenly, slightly we have increased the dose. There was huge changes we are seeing in the way we are seeing the very significant toxicity. What could be the reason? So this could be due to the region of the supraproportional increase of the plasma exposure with the dose. This is possible. We can see this kind of effect when we are increasing the doses, because if in case the exposure inside the you can say the plasma concentration increasing more than the dose increase, then it could lead to the over kind of a toxicological effect, which may not be seen at the lower doses. So if you have the kinetic understanding, you have the relationship of dose or exposure, you can understand what might be happening to the in vivo scenario. It is also quite possible that no evidence of toxicity is observed even at maximum technical feasible doses. This is also very common phenomena is seen that when we try to do the toxicological studies and we give the doses, we may not see any toxicity, and we may, if we do not understand the toxicokinetic, we may interpret that the compound is not toxic and we can recommend it for further studies. But it is quite possible that the dose which we have used in the toxicology study, at that dose, the compound might have not absorbed. It might have not reached to the systemic circulation, and it is quite possible that because of that reason, we are not seeing any effect. So for that reason also we need to understand what is happening in the concentration with increasing the dose. So as you know that dose response relationship, which is a very simple concept. In general, the greater the amount of a substance that enters your body, the greater is the effect on your body. So this connection between amount and effect is called the dose response relationship. So which is most of the time is available. But to interpret this dose response relationship, we have to understand the dose concentration relationship. And that is how when we combine dose concentration relationship versus dose response relationship, this is called the PK-PD or TK-TD. So what could be the fundamental reason when we say that when we are increasing the doses and we are seeing the saturation in the kinetics, that exposure is not increasing to the same extent or increasing more than the dose increase. So these are the primary fundamentals, 4 parameters you can always look into that. The gastrointestinal, which is the major first site where the compound is getting absorbed. So it is quite possible that the solubility could be the limiting factor. As you are increasing the doses, the compound solubility is very low, compound is might getting precipitated out, and you are not seeing increase in the exposure as the dose is increased. Similarly, it is quite possible that some certain kind of transporter which are helpful into the intestine for absorption of the compound might get saturated, and those can affect the absorption process of the compound. Again, the distribution can also be affected because of saturation of the binding sites of the plasma protein binding as well as the tissue binding sites. Renal excretion is also like a tubular secretion, especially which is there are different kind of transporters which are involved in the excretion of a compound from our body into the urine. So it is quite possible that at the higher concentration, this transporter may get saturated, and our body is not able to eliminate those compounds from the body, and the concentration is increasing, and it may increase the toxicity. And another very important aspect is the hepatic metabolism. As you must be knowing very well that liver is the primary site of metabolism. If the compound concentration is increased to a significant extent into the liver, it is quite possible that it may saturate the enzymes which are involved in the metabolism of a particular drug or chemical. And if its metabolism is not happening further, that concentration will keep on increasing, even with the more than dose proportional increase you may see into the systemic circulation. So, when we talk about that drug molecule, what we expect from a kinetics perspective, what should be the ideal ADME property of a molecule? So, as you know that always one of the preferred route is the oral route. So we always wanted to have a compound or a new chemical molecule which can be administered by the oral route. And for oral route, we generally prefer that the compound should have good solubility and permeability. Okay. And we also want a compound with minimal first pass effect and good bioavailability. I will discuss in detail that what is the first pass effect and bioavailability. And you must be knowing that the bioavailability is one of the fundamental parameter which is responsible for any compound, like when we administered and how much is getting absorbed into the systemic circulation become very important. Metabolically stable, but not too stable. So this is again very important. Let's say if compound is getting very quickly metabolized, so this is also like a disadvantage situation, because if it is getting too fast metabolite, it means that I have to administer too much higher dose to achieve the therapeutic concentration of the compound inside the body. So we prefer the compound which is like comparatively stable, but not too stable. Again, linear kinetics, we expect that compound should have the linear kinetics, because if it has the non-linear kinetics, then it becomes unpredictable in terms of the kinetics of the molecule, and we do not want to take any molecule further for the clinical trial studies which we do not understand the kinetics of the molecule. Another important aspect when we talk about the kinetics perspective is the balanced clearance, because we do not want to have a compound which is only metabolized or only cleared by a single route, because of any drug-drug interaction or any other problem, if that route got blocked, it is quite possible that that compound might get accumulated inside the body and you may see the toxicity. And we also wanted to have this not an inhibitor or inducer of ADME enzymes. Let's say if my molecule which I am trying to develop as a drug is an inhibitor or inducer of the certain enzymes, so it's quite possible that it could affect the kinetics of other molecules which are co-administered with the drug. As you know that we take several medicines together at the in the like different disease conditions. So if they interact with each other, it's quite possible that it will affect their kinetics and it will affect their therapeutic efficacy, or in fact it could lead to the toxicity. So we prefer the molecules which are like not an inhibitor or inducer, but their compound could be always be some inhibitor or inducer of that. We have to take the decision, but we prefer the molecule which are not inhibitor or inducer of any enzymes. And the wide therapeutic index, as we have seen in some previous slide, that we wanted to have a molecule which provides us a larger window of the toxicological effect concentration versus therapeutic effect concentration. If this gap is larger, it means that my safety window is very big for my molecule, and it is like quite good for the drug, that it will not show the toxicity because of the very wide therapeutic index. So what actually happened for any drug molecule when it is being administered? So as you can see that this is I have included for any other toxicant or phytochemicals or any drug molecule. So after the absorption process, the compound goes into the plasma and it exists in the two form, either free form as well as the bound form. So we have to remember very important concept that only the free drug is available for either for the metabolism or elimination from the body, or at the same time for the any kind of therapeutic effect. Because the drug which is bound to the protein will remain into the systemic circulation, and only the free drug will distribute into the different organ. And because most of the time our site of action is like outside of the blood, so compound has to distribute to reach to the specific site to exert its effect. So the understanding of the kinetics from the kinetic perspective, plasma protein binding become very important, very crucial for any given molecule. So as you know that I talked about that how especially in terms of the dosage regimen, when we say that how much and how often, or a drug chemical we can take or any drug molecule we can take. So there are some fundamental parameters which are very important: volume of distribution and clearance. Remember, these are the two very fundamental parameters which we need to understand, because they govern the several other parameters of the body, especially for any given chemical. The clearance will directly affect the bioavailability of a molecule, as you can see, and so bioavailability is affected, then affecting the systemic exposure. So then it is affecting the how much that drug we have to take. Similarly, clearance will have direct impact on the half-life of the molecule, and volume of distribution also have the half-life effect on the half-life, and this will determine that how often we have to administer a drug chemical, because half-life will determine that how fast it is getting remaining of half of initial concentration, and based on that, based on understanding the clearance and volume distribution, this will help us to understand that how much and how often a drug chemical can be given.
Due to some network issue, wait for just 2 minutes. There is some network issue. Please wait for 2 minutes.
Yeah, Vaibhav sir, good afternoon. What are we waiting for now, sir?
Sir, some network issue from Dr. Singh. Just wait for 2 minutes.
Okay.
Dr. Vaibhav, I'm sharing my screen again. Okay.
Please, sir.
Yeah. Okay. So am I audible now?
Yes, sir.
Yes, sir.
Okay. So as I was talking about, there are two kind of bioavailability when we talk about. One is the absolute, which is like a property of any given molecule when we try to compare with the oral versus IV or any other route of administration. And another one is the relative when we do not have the, let's say, IV data and we try to just wanted to compare to different kind of formulations. So at that time, that comparative assessment, especially from the bioequivalence perspective, we call it the relative bioavailability. So another very important fundamental parameter is like what is the difference between the absorption versus bioavailability? Okay, well, sometimes people get confused that bioavailability and absorption phenomena. So absorption will just only tells us the movement of the drug from the site of admission into the blood, which drains the site of administration. Okay, so this is simply the absorption process. As you can see in this particular figure, that how that in that some capillaries which is coming to the intestine, taking out that drug molecules into through the liver. And bioavailability refers to the amount of drug which actually gains to the access to the systemic circulation. So when we talk about the systemic circulation, it means that we are talking about the circulation in the arterial circulation. It means that drug which is reaching into the systemic circulation after the pumping of the blood by the heart. Okay, so after the oxygenation of the blood, the blood which is being pumped by the blood which is pumped by the heart to the body is called the systemic circulation, especially the arterial circulation. Okay, so the bioavailability is primarily defined based on the systemic circulation concentration in the arteries. And before that, whatever is happening with the molecules is we'll call the first pass effect, and which is responsible for reducing the bioavailability of any given molecule. As you can see that in this particular figure, when any compound is being administered, there are the primary parameter which will govern first is the solubility and permeability of the compound, because any compound which has to be absorbed first come into the solution form. and how it will come into the solution form, it will be the solubility which will determine it. So the solubility at the same time, if whether compound is stable or not, is also very important aspect, because compound has to be stable in the gastric environment or intestinal environment, because then only it will get absorbed. If it is not stable, that will contribute to reducing the bioavailability of the compound. So once the compound is solubilized, then next parameter will come, very important parameter is the permeability of the molecule, because once the compound in the solid form, it has to permeate through that intestinal membrane, because if it is not having the very poor permeability, then it is again going to have the poor bioavailability, because if the compound is not getting absorbed, then what will happen? It will go directly into the feces in the unabsorbed form. So that compound which is not absorbed is going directly to the feces, and so this will contribute into the reducing the bioavailability of the compound. So three important parameters which are responsible for determining the bioavailability are the fraction Fa, which is called the fraction absorbed. Fg is called the fraction escaping the intestinal metabolism. Fg is called the fraction escaping the intestinal metabolism. And FH, which is called like the fraction escaping the liver metabolism. So if we understand that Fa, Fg, and FH, we can determine the bioavailability of any given molecule. So the primary site of metabolism is liver, but before liver, the intestine can also metabolize the compound. So that is why it's the compound which is like when passing through the intestine and not getting metabolized, that fraction is called the Fg actually, that unmetabolized fraction passing through the intestine. And then it goes to the portal vein, and portal vein carries that molecules coming out from the intestine to the liver, and then liver will metabolize the compound, and the compound which remain unmetabolized is called the FH. So FH is a fraction escaping liver metabolism, and this amount actually goes into the systemic circulation. So that is why if we understand Fa, Fg, and FH, if we multiply these three components together, we can calculate the bioavailability of the compound. This is a very simple like example again in terms of how that phenomena is happening inside our body. Like any compound which is administered, it goes to the gut, then again it goes to it is the gut wall, which is called the fraction absorb. Okay, and this all will take to the through the liver, then compound goes to the through the vena cava, it goes to the heart, and then heart it goes to the lung. Sometimes what happens, certain molecule can also get metabolized by the lung also, but they are very minimal actually. But it is quite possible lung can also contribute to the metabolism. So if this is also contributing to the metabolism, then another fraction will become important is Fp. Okay, fraction escaping the first pass metabolism by lung. So Fa, Fg, Fh, and Fp together, if we know, we can calculate the bioavailability of the molecule, because the compound which is coming out from the lung, remaining compound coming out of the lung and reaching into the systemic circulation, which is called the arterial circulation, is determines the bioavailability of the molecule. And these all parameters which we talk about, Fa, calculated by some in vitro test system also. So that is why I'm highlighting, even if like we do not able to conduct the in vivo experiment, we can do in vitro experiment to determine, to utilize the understanding of the bioavailability concept. This is again in terms of the amount I have explained this, how the bioavailability being calculated. Let's say you have started with the 100 molecules and 20 molecules remain unabsorbed. Okay, so the fraction absorbed was the 0.8 of that molecule. So of that molecule, 80 molecule reached into the portal vein, okay, into the liver, and then let's say 20 molecule got metabolized. So this is giving you the fraction escaping the liver metabolism 0.75. So like that, if you understand all the components, you can multiply it together and it can give you the bioavailability of 0.3. 0.3 nothing but it's a 30% bioavailability of the molecule. So you can calculate the simply bioavailability by understanding Fa, Fg, and Fh of a molecule. But if you suppose you wanted to calculate the bioavailability of a molecule by in vivo study, then what should be done? So for the in vivo study, we have to always compare with the IV, because IV route will give us that 100% bioavailability of the molecule, because we are giving drug directly into the blood circulation. And then we can compare the exposure IV versus with any other extravascular route, and we can calculate the bioavailability of the given molecule by using this simple formula. So some examples, the in vitro test studies, which are like we generally regularly conduct for any kind of pharmaceuticals molecule when we are doing the preclinical studies. So certain very important absorption studies are the solubility and stability studies, PEMPA studies, and CACO2 permeability studies. These are the, as I told you in the previous slide, that solubility and permeability are the, and stability. These are the three fundamental parameters. The first step itself will determine your compound absorption. So these we can, so for determining the absorption, we have to conduct these three studies. As I told about the distribution, the binding is important, then we have to determine the plasma protein binding of the molecule. As I talked about that metabolism, because it is responsible for the clearance of the molecule. So we can use the microsomes, liver microsomes for metabolism study. We can use the hepatocytes, and then we can do the in vivo correlation to understand the clearance in vivo system. And another very important studies class is the CYP phenotyping and invasion study. CYP phenotyping and invasion is nothing but we wanted to understand what are the different isoforms of the cytochrome P450 enzymes are involved in the metabolism of my chemical of interest. At the same time, I wanted to know whether my chemical of interest will affect the metabolism of other molecules or not. Then for that purpose, I have to do the invasion studies of that particular these enzymes. So why these are important? Because when we take any tablet capsules, this is the process which happened to bring any compound into the solution form. Okay. And to understand compound solubility or stability, what we can do, we can take, these are simple in vitro studies which we can do in the laboratory. We can use a simulated gastric fluid. We can use a simulated intestinal fluid. The composition, you can easily get it into the in the pharmacopoeia. You can simply you can make the form like a simulated fluid, and you can do the stability study. If your compound is stable in these pieces, at least you can see that your compound is not going to have the bioavailability poor because of the stability issues. Similarly, you can determine the solubility of the molecule in the different buffer, this gastric fluid and intestinal fluid, to understand whether your compound is having the good solubility or not, so that it can be absorbed from the intestine. So these are the, as I talked about the permeability studies. So in vitro cell culture, there are different assays which we can use for this purpose. As I told you, the PAMPA assay. These are the simply 96 well plate, which are easily available and can be purchased, and directly we can use for any kind of screening. And major advantage, these are very high throughput, and there we have the artificial membrane which is made of the lipids, and we do the permeation assay. Similarly, the SCACO2 and MDCK are the cell culture based techniques, where we need to have a cell culture facility inside the lab for conducting such studies. Another important aspect is averted intestinal segment. This we can do again in the laboratory easily, because if we have the animals, we can take out their intestinal segment and we can do the study of permeability studies. Another important is the in situ intestinal perfusion system. This we can do directly into the animals, where we can cannulate the intestinal portion of the rat or any other animals, and we can do the perfusion studies. And then we have the simple formula. If we have calculated the permeability, this is called the P apparent. The P apparent value we can get from these different experiments. Then by using this simple formula, we can calculate the fraction absorb. So as I told you that three things which are required: fraction absorb, fraction escaping cutward metabolism, which is FG, and FH. So FA you can calculate by these experiments. For FG, again, you can use the intestinal microsomes for metabolic stability. And based on that intestinal microsomal metabolic stability studies, by using simple these formula, you can calculate the FG of the molecule. So like why we have the different kind of assay, right? This PAMPA we have, CACO we have. What are the advantage of having this particular different kind of assay system? So what is the major difference in CACO and PAMPA is simply is an artificial membrane. It doesn't have any kind of active process, right? This is completely a passive process. It means the compound, whatever is having the lipophilicity, it's getting perfused through the membrane. Whereas CACO is like simply the intestinal cells based system, where all the active and process paracellular, or you can say the active and passive process exist. So if you wanted to have the mechanistic understanding of your molecule, whether my molecule is like passively absorbed or whether it is actively absorbed, whether it is like absorbed by the active as well as the passive process, whether any kind of influx transporters or efflux transporters are playing any role in the molecule's absorption. By having these two data, we can always interpret the data in terms of the molecule permeability. This is another simple example that how the CACO2 study is conducted. This is simply, you can do the 24 well plate kind of system you can use, where one is the donor side, one is the acceptor side, or this simply one is represent that intestinal side, another side, basolateral side represent the blood side. So you can put the compound in the apical side and measure the compound, how much amount is coming into the basolateral side. Similarly, you can put the compound on basolateral side and measure the compound, how much is coming to the apical side. And then using the simple formula, you can calculate the permeability of the molecule. So this is the simple the classification of the molecules. Like let's say if somebody asks you that whether your molecule is having the poor permeability or good permeability, how you will decide? Okay, so these are the certain classification which you can use depending on the how much permeability of your compound you got from the in vitro experiment. And based on this permeability number, you can define whether your compound is a poorly permeable or high permeable molecule. And you can see that this is a very simple, just I have given you the example here, that permeability, how well is correlating with the bioavailability of the molecule. This is the in vivo study data which conducted in different species, hamster, rat, and rabbit. And you can see that compound is having a very poor bioavailability. And when we did a lot of mechanistic study, we came to know that that low bioavailability was because of the poor permeability of the molecule, which is 1.1 into 10 power minus 6. So this comes under to the low permeable class of the molecule. So this is how you interpret the data when you have the different kind of molecules data understanding. This is the setup of the PAMPA plates. This is again very much similar to the Keiko plates, which I have shown you, but this is a 96 well plate system, where again, this also has the two side acceptor as well as the basolateral size, and you put the compound in the acceptor side and you measure into the receiver side, and then you calculate the permeability of the molecule. This is the in situ perfusion. This is in situ perfusion assay, where you, as you can see, this particular animal, we anesthetize the animal, and we cannulate that intestine from one side, and we perfuse that intestine by simple peristaltic pump. We supply the fluid, and we measure that fluid out, and then we measure the concentration in versus concentration out. So the difference in the concentration, which was in versus out, will help you to calculate the permeability of the molecule by using this simple equation. This another example which I told you about, the inverted averted gut model, in which this simply we just cut that in this is the difference between that in situ versus this one. That in situ the animal was intact, actually it was only anesthetized and surgery was done, whereas in this particular experiment you are completely taking out that intestine, cutting into the two pieces, and ligating this in both the end and putting the solution in outside of this particular let's say in the petri dish, and you have averted the intestine. So inside out, what you did, the inside part came out and outside part gone inside. So as you can see that villi out of this, these are the some membranes, these are the villi of that particular intestine which came out. So when you are putting this particular segment into the buffer which contain the drug will get absorbed and reach into this particular pocket of this averted gut. And then at the end of the experiment, you can take out this intestine and you measure the concentration inside this sac of this particular intestine, and then you can calculate the concentration which was present in the, let's say, the petri dish, and how much concentration came inside this particular sac, and then you can calculate the permeability of the molecule. Coming to the this volume of distribution, that how we can determine, as I told you that the volume of distribution, again, very important parameter, because it will help you to understand that how much amount of drug is free in the systemic circulation at any given time. So this is a simple like a equilibrium dialysis experiment which we can conduct, where we divide that well into the two part. One part is called this divide we do by using the semi-permeable membrane, which will only allow the free drug to pass. It will not allow the bound drug to pass from one side to another. Okay, so one side you have the buffer which doesn't contain the drug, and another side you have that let's say the plasma where you have spiked the drug. And after particular hour of the, let's say the 4 hour or 6 hour, you can measure the concentration both the side, and then you can calculate the plasma protein binding. And how we can do this is, these are the very simple device which are available, which we can purchase, that rapid equilibrium dialysis device. These are ready-made device which already contain that membrane also. You have directly use this membrane, and directly this 96 well plate you can use for your experiment. And another device is the ultra filtration device, where you can put your sample on the above and on this tip, and then you can centrifuge this, and only the unbound drug will settle down. It will came in the filtered fraction. Unbound drug will come, and you can measure the concentration in above and the down filtrate, and then you can determine the protein binding. So why this is important? Because this directly affects the distribution, volume of distribution, and volume of distribution is again very important to know that like I should know whether because as I told you that the volume of distribution and clearance both governs the half-life of a compound. So if compound is having a very high volume of distribution, it is going to increase the half-life of a compound. So how to then, another important aspect is how to interpret that volume of distribution, whether it's a low volume of distribution, intermediate volume of distribution, or high volume of distribution. This can only you can correlate with by understanding of the physiological volume of the body. So let's say the plasma has the volume of 3 liter, extracellular fluid is 18 liter, and total body water is 42 liter. So in terms of liter per kg, you can always interpret, let's say the 0.6 liter per kg is our total body water. If your volume of distribution is coming more than this, it means that your compound is getting significantly distributed and compound is going to have the high volume of distribution. So this is how you can correlate your number from the in vivo experiment volume of distribution, and you can interpret in terms of the physiology of our human body. So coming to some already which I explained you about the different experiment. So coming to what are the different test system and how we derive this test system for metabolic stability. Okay, so we understood the absorption part and we understand all the experiment. Now I'm coming to the metabolic stability, because this is another important parameter which will govern your compound clearance, and the clearance will directly govern the viability of the molecule. So you can always prepare this particular setup. We can do in the laboratory also easily at the academic level, where we can homogenize the liver and we can prepare the different fraction, S9 fraction, cytosolic fraction, macrosomal fraction, and these different fraction has different kind of enzymes. So selection of the test system is also very important, that which particular test system we are using. Hepatocyte is like an ideal preference because it is a very intact system. It has all the enzymes at one place, whereas S9, cytosol, and macrosomes are easy to use and easy to handle, but they have certain different enzymes depending on the fraction which we are using. This is a simple setup that if you wanted to do the metabolic experiment, how to do this experiment, you can just take any kind of like enzymes or fraction, and then you put your test compound. Then you can start the reaction by adding the cofactors. Again, this cofactor depend on like which particular enzyme you are using. Okay, so and based on that by metabolic state, and then you can collect the time different time points your sample, and then you can measure your compound concentration. So if compound concentration is degrading with the time with that particular enzymatic system, it means your compound is getting metabolized by that pathway, and then you can calculate the clearance of the compound. This is a simple assay condition I have put into slide. In case if you wanted to use this particular assay, what should be the ideal assay condition? So I have mentioned here all the ideal conditions which we can always use for different kind of metabolic stability experiment. And even in fact, I have mentioned certain kind of that cofactor which are also being used for different experiment. NADPS is one of the most commonly used like cofactors for the cytochrome P450 enzymes mediated metabolism, and UDPDA is primarily used for the gluconide UGT enzymes metabolism understanding. So different cofactors are there, and let's say if you wanted to know the role of sulfotransferase enzymes, you can use the PAPS as a substrate, as a cofactor. So different cofactors are used for different enzymes. This is the protein concentration, how much we should use for experiment. Liver macro, generally people use 0.5 mg per ml. Liver S9, generally bit higher concentration are being used. If suppose I wanted to use the hepatocyte, then how much of the cells I should use for metabolic stability experiment? If suppose I am using the recombinant enzymes, then how much concentration I should use? So these are the certain concentration range which I have given, which you can use for the different experiment. And another important aspect is that how much concentration I should put on my test compound in the metabolic stability experiment. So again, it is better to use as much as low possible, because we do not want to saturate the enzymes when we are doing metabolic stability experiment. So you can see that how much is like a correlation between the metabolic stability versus in vivo performance of a molecule. As you can see in this particular figure, that drug A versus drug B, you can see that drug B is getting metabolized very fast. You can see that percentage remained of a parent compound in metabolic stability experiment within 30 minutes, it gone almost completely, right? But as compared to the drug A molecule. So when we did the in vivo study of these two molecules, what we found that drug A versus drug B, drug A is having a very good viability, 62%, as compared to drug B, which is having only 7.9% viability. So this is how you can correlate the data. That is why before going to the in vivo experiment, we do the in vitro experiment, so that any molecule which have the poor property, we can eliminate them there itself, and we do not take those molecules further for the development purpose. This, and if suppose you wanted to calculate this, if let's say if you have done your experiment in the like laboratory, and then how to estimate and how to calculate this half-life, this also I have put into the perspective. Let's say you have taken the time and different concentration, you have measured the compound, then you can plot it with the time versus natural log of the percentage compound remaining, and then you can get the slope value from this. And that slope value is nothing but the elimination rate constant, and from that simple formula T half equal to 0.693 upon KEL, you can calculate the half-life of a compound. And from this again half-life, you can calculate the compound clearance numbers by using this again formula, which is at the bottom, like 0.693 upon in vitro T half into ml of incubation divided by mg of microsomes. So this is again very important because we are using in vitro test system. So how much protein we are using, that also we have to bring in into the clearance number calculations. This is again just to give you a little bit understanding that if suppose one can ask me that we are using the in vitro experiment, but in vitro system are very different from the in vivo. Okay, and if anything which is done in vitro, how it will be translated to the in vivo. So whatever the intrinsic clearance we calculate in vitro, we translate to the organ level and to the body level by using the different scaling factors. So you can see that this particular microsomes equation and hepatocyte equations are given here. So whatever data I am doing into the small protein, let's say I have used only 0.5 mg per ml protein, 1 mg or hepatocyte cells number, but liver may contain a very high number of cells. But then how to translate that data from in vitro to in vivo? So that understanding, we already know that per gram of liver, how much of the cells are present, per gram of liver, how much of the microsomes are present, and what is the liver weight per kg of the body weight. So by bringing all these information together, we can convert that in vitro data to the in vivo level. So this must be, you must have read in like when we do, when we study in the like B farm or even M farm, that any compound which is going in and compound coming out of the liver will help you to understand how much compound got metabolized. So, and whatever compound got metabolized, we call it the fraction metabolized, and we call it as the extraction ratio. So you must be knowing this terminology, extraction ratio. So this is again calculated based on the clearance number. Okay, but remember that clearance and that hepatic blood flow of that particular organ will help you to calculate the extraction ratio. Why? Because what could be the maximum capacity of any given given organ to metabolize a compound? That will be that availability of a molecule. Let's say if my rate, and that will depend on the blood flow. Let's say my blood flow is 10 ml, let's say 10 ml per kg, and my compound clearance is also the same rate. It means that compound, whatever is coming in at the outside, when it will come out from the liver, I will not see any compound because it will get completely metabolized. So this is how, so the maximum capacity is like the availability of the molecule, and that could that will depend on the hepatic blood flow. So what could be the maximum capacity of metabolizing for particular drug is that maximum hepatic blood flow. So that is why I have mentioned that hepatic blood flow of different species at the bottom, because this will help you to interpret the clearance of a compound, whether my compound is a low clearance compound, whether my compound is an intermediate clearance compound, whether my compound is a high clearance compound. This you can correlate with the hepatic blood flow. If your clearance number is close to the hepatic blood flow, it means your compound is high clearance. Okay, so accordingly, you can understand the clearance of a compound, and accordingly, you can calculate the bioavailability, rough bioavailability, you can calculate by simply understanding the clearance and the hepatic blood flow. See this particular example I have put into the here, extraction ratio, you must be knowing that when we call the compound having a extraction ratio more than 0.7 is a high clearance compound and less than 0.3 is a low clearance compound. So this is like how this particular number is given for a one compound for 4.8, 6.4, 1.5 ml per minute per kg in rat, monkey, and dog. Okay, this is the clearance number. Now, point is that now I'm asking you that, okay, whether this compound is a low clearance compound or high clearance compound. Okay, so we will say this compound is low clearance because if you see that the rat hepatic blood flow is 55 ml per minute per kg, but my rat clearance is only 4.8 ml per minute per kg. So it means my compound is a low clearance compound. And how I have decided this? Because 55 is the hepatic blood flow into the rat. Similarly, in the monkey is a 44, and let's say in the human is a 21 ml per minute per kg. If I can in vivo data or either whether in vitro data, I can correlate this clearance number with the hepatic blood flow, and I can tell whether my compound is a low clearance compound or high clearance compound. So coming to some in vitro, some or other important aspect again from the phenotyping perspective, as I said that because when you see that what will happen when a single, let's say you have a drug molecule which is a victim, okay, and when you administer this drug with another drug molecule, it is quite possible that they could interact each other, and drug B may influence the kinetics of the drug A molecule, and it will lead to the accumulation of that compound, right? So that is why it is important to understand what are the different enzymes are involved in the metabolism of a particular compound, so that I can understand whether my drug is going to have any kind of interaction with another drug molecule which is co-administered with that. So how I can do that. Okay, so there are different enzymes which are recommended by the US FDA, and these particular enzymes are the mostly responsible for metabolism of most of the compound till date, whatever available in the market. So CYP1A2, CYP2B6, 2C8, 19, 2D6, 3A4. These are the all important enzymes which we have to screen for any given drug molecule. Okay, we have to know whether my compound is a substrate for these enzymes, or whether my compound is inhibiting these enzymes. This both information I should know. So, and when I wanted to do the inhibition experiment, what would be my aim? My aim will be to calculate the IC50 of my molecule, to calculate that inhibition rate constant. And if I understand this inhibitory concentration, then I can understand whether my drug is going to have interaction with any other drug or not. This is a simple classification. Like if you know your compound Ki value or IC50 value from in vitro experiment, then you can calculate whether it has any chance of in vivo interaction or not with any drug, any inhibitor or any other drug molecule. So coming to the reaction phenotyping, so reaction phenotyping is nothing but it tells you about that particular what are the different enzymes which are responsible for the metabolism of your drug molecule. Okay, so the primary purpose of reaction phenotyping is to identify the enzymes which are responsible for the molecule metabolism. And what we prefer in terms of the understanding of this metabolism by different enzymes. As you can see that this first example, you see that one, let's say my compound is metabolized by only one enzyme. Okay, what will happen? Let's say if this particular enzyme is inhibited by another drug molecule which is co-administered with that molecule. So this will call the drug-drug interaction. What will happen? Because there was only one pathway for elimination of that molecule, and that also got inhibited by another drug molecule, its concentration will increase significantly into the blood, and it could lead to the toxicity. So I do not want to have a molecule which is having metabolized by a single enzyme. I would prefer compound which is metabolized by either two or many enzymes, so that even if one enzyme is being inhibited by another drug, other enzymes are available to take care of the metabolism of that particular compound. So this is what we expect from our molecule, that it is metabolized by multiple enzymes. And what are the different way of calculating it is? Again, they are different. You can use the recombinant enzymes for this purpose. You can use the microsomal test system, where you can use different specific antibodies to inhibit a specific enzyme, and then seeing whether your compound metabolism, what is happening to that. And certain kind of correlation analysis also we can do, where we have the human liver microsomes from the different kind of population which are deficient in metabolism of a particular enzyme. Okay, so like this different test system we can use for understanding of different enzymes, or at least two systems we have to use for making a decision on what are the different enzymes are involved in the metabolism. So coming to a very simple now it's kind of case study, though it is not a one-on-one discussion in that sense. But if you see that why I have put this particular data is, let's say I have done, this is how we apply that understanding of kinetics. Let's say I did the kinetic study of a molecule, I did the intravenous study, I did the oral studies, and I came to know that my molecule is having a bioavailability only 6.3%. Okay, but my compound clearance is only 4.6 ml per minute per kg. And remember that hepatic blood flow was 55 ml per minute per kg in the rat. So compound clearance is 4.6 only. So it means compound is very low clearance compound. But still this compound is having a very low bioavailability. So what could be the reason? We have to find out, because in the kinetics, is very important thing is that in the case of drug discovery, we have to know the each and every reason that why the compound is having low bioavailability, why this compound is behaving abnormally, though it's compounding like a stable molecule and still not having the good bioavailability. So we have to understand the reason, because if we are not having the understanding of the reason, we may fail in the clinic. So that what we did, we determined. So as I told you that compound bioavailability can be affected by solubility, stability, permeability, and clearance. Okay. So what we did, first thing what we did, we determined the solubility of the compound. From the solubility experiment, we came to know that the solubility of the molecule is very low. So we thought, okay, solubility could be the factor for its low bioavailability. We could not determine its permeability because solubility was so very low. So we couldn't do the in vitro experiment for permeability experiment. But we did the clearance experiment, that we know that compound is a low clearance compound. So the clearance cannot be a reason. So the reason could be either solubility or permeability. So from the solubility experiment, we came to know the solubility is low. So what next I did? Then I made a solution formulation. Instead of suspension, I used a solution. The reason for making solution was that I wanted to remove the solubility limitation first, because I wanted to know whether it's a solubility or permeability. So I made a formulation which is like already my compound was soluble in that, and I administered again. I did the study, and I did again the bioavailability assessment with the solution, and you can see that bioavailability again was the same. Okay. So question was then what is like a factor which is responsible for low bioavailability? So the factor remain is the permeability. So this is how we make a decision, like in terms of interpreting the kinetics or interpreting the toxicokinetics data, or making the decision by using the different in vitro experiment and different in vivo experiment. So that's it from my side. I would be happy to take any questions further.
Thank you, sir. Thank you, sir, for valuable information in toxicokinetic and pharmacokinetic studies. Routinely, we use different techniques in pharma academic research and in pharma industry, also in toxicology research. So, any question from participants? Sir, I have received one chat box question. They are working. They are working in, I think, plant-based drugs. Their question is how much initial dose were to take during the animal studies for acute toxicity studies?
For acute toxicity study.
Yeah. Their starting material is a plant extract. For acute toxicity, how much dose were to take or select?
So my Dr. Akshay Dwarkar already actually explained that how the toxicological doses are being selected initially, especially acute toxicity study. But from my perspective, what I would tell them, where first thing is like we should know what is my therapeutic dose. Okay.
Yes.
So if you know the therapeutic dose, then from the therapeutic dose, I will start estimating my doses for the acute toxicological studies. So my start dose will be from the therapeutic dose I will start, and then I will increase, let's say, the 5-fold or 10-fold, and then I will go into the toxicological study. According to that, I will have to decide the doses for acute toxicological study. So very first thing is that you should know what is the therapeutic dose for your molecule.
I think also if you refer the OECD guidelines, they mention the initial dose.
Correct. Yeah.
So minimum dose and maximum dose.
So before that, we should know the kinetics and the therapeutic efficacy of the molecule, like therapeutic dose. We understand the dose and concentration relationship there. Then we can decide the doses, because our aim should be in the toxicology studies that my compound should be safe at least 10-fold exposure of as compared to the therapeutic exposure. Okay, so target should be at least 10-fold. And if at the 10-fold I am safe, my compound is safe in the animal studies, then at least I am sure that my molecule is not going to behave abnormally in terms of toxicological in let's say the advanced studies.
Yes, sir.
So if I may just add further to what Dr. Shilendra said. So once you know the therapeutic dose and you have some PK PD data, and if you look at OECD 420, then you can use that and you start from say like 10 times of your therapeutic dose. But if you're looking at 423, then you look at if you know that I mean you have information on therapeutic dose, you can start with either 2000 mg per kg or 300 mg per kg, depending on what is your therapeutic dose.
Yes, sir. Thank you, sir. Sir, other question is which is the best study, in vivo or in vitro study, and how to induce disease in vitro methods? So
again, I would say very open question. This is depend on the like an endpoint which we are interested. Okay, so nowadays even we are hearing a lot about that we have to ban the animal uses for different experimental purpose or different even toxicological studies or other kind of things. So more reliance we are going to the in vitro test system. So depending on the endpoint which we are interested, accordingly we have to decide the in vitro test system. Okay, let's say if I wanted to do the understanding on effect on the brain, so I have to use the neuronal cell lines, different kind of cell lines to understand the molecular pathway which are being affected. And let me tell you that nowadays a lot of research is going into the direction of the in vitro model validation and trying to understand the finding to the human being or directly prediction to the in vivo scenario. So again, like those who are doing any kind of in vitro experiment, they have to have understanding of the kinetics. Without understanding of the kinetics, we cannot translate in vitro toxicological data or therapeutic data to the human being. So So now the kinetics component is again going to come much like in the bigger perspective, especially for translating the in vitro findings to the in vivo, because in the coming years we are going to see completely ban on the animals.
Any other question, please? Thank you, sir.
Thank you, sir. Thank you so much, Dr. Vaibhav, for giving us opportunity, and it was really nice to interact with such a huge number of students, and it was always good to interact with the students, especially I would say.
Yes, sir. Thank you, sir.
Thank you.
Now,
Thank you.
next speaker is Major Retired Dr. Jitendra Singh. Sir is presently working as a head and professor, Department of Pharmacology, Maharashtra Postgraduate Institute of Medical Education and Research at Nasik. Dr. Singh having more than 25 years of experience in the field of medical pharmacology. Dr. Singh also involved in various clinical studies project national level. Dr. Singh also worked in medical division of Indian Army. Also recently, Dr. Singh received the highest number of publication in Index Journal at National Association of Pharmacology and Therapeutics, that is a NEPTICON conference, which was held at Lucknow by Department of Pharmacology and Therapeutics at King George Medical University, Lucknow. Now, I request Dr. Singh to proceed the sessions for scope and potential of pharmaceutical medicine. Please, sir.
Thank you, Dr. Vaibhav. Am I audible to you all?
Yes, sir.
Okay, so thank you so much, Dr. Vaibhav and MUHS for giving me this opportunity to interact with the students. I was listening to the vibrant sessions which happened in the morning, and they were really good. My compliments to both the speakers for a wonderful session which they have taken. And now I just want to say to the students that they've seen or they've heard a lot about pharmacokinetics, but now I'll be talking about the practical aspect. It all starts with absorption and distribution. So once you complete your degree, now you are you are wanting to get absorbed in the system and to get distributed to your site where you really want to work, be it pharmaceutical industry. So what I mean to say is absorption and distributions are not only principle of pharmacokinetics, but they also hold very true when you are looking for a varied career options in your life. MUHS has already started a course of MSc in pharmaceutical medicine. This is running for quite some time in MUHS. And now, like initially, it was a 2 years course from, yeah. Sir, am I audible, sir? Vaibhav, sir, am I audible?
Yes, sir. Yes, sir.
Okay. Yeah, so it's been running for quite some time. And this MSc in pharmaceutical medicine course is conducted in Maharashtra University of Health Sciences. It's a UGC approved certificate course, and it is the format of this course is blended mode, right? You are doing the on-site sessions mainly at Nasik, so the contact sessions is done at Nasik, and remaining, the teaching part happens online also. So it is conducted in blended mode. And like there are various career avenues which a person who is into pharmacology or pharmacy is looking for. Like if you do this course, you open up your career avenues in a pharmaceutical industry, which is one of the dream jobs for most of the people who are working in this particular field. Nowadays, pharmacovigilance is also coming up in a very big way. We have got one entire module of pharmacovigilance which we taught in this course, where a lot of hands-on experience is also being given in terms of generating of the ADR reports, how the PSURs are being updated, and how you report it online also to the Uppsala Monitoring Center through various channels. You can also get, or you can also work like a research officer or a regulatory officer, medical writing, or the second part is if somebody is really ambitious in joining the ClinOps operation. So like if you want to work as a CRC or a CRA or clinical data manager. So this course teaches you a lot about, hands-on handling about how the clinical trials are being conducted, how the protocol is being written, how the ethical concerns are being taken care of, and how you proceed in a particular trial. So it definitely makes you fit to join a CRC or a CRA. Nowadays, like clinical data manager is a very important post which is coming up in various center of excellence, which is coming up at various different places in Nasik. And I am happy to announce that even MUHS is coming up with a center of excellence, which will be functional very soon. And ClinOps appears to be one of the important pillar which will be there in the COE, that is Center of Excellence. So basically, if you are doing this particular job, sorry, I mean this particular course, it opens up your avenues to join a pharmaceutical industry or in clinical trial research or as an ethics committee member also. Now that ethics committee is a very important pillar for conducting academic trials. You need ethical committee approval. Nowadays, you don't need any approval from the regulators till the time you are not interested in getting the drug into the market. If it is only for academic purpose, the ethics committee can give the permission. And once you are when you are taking such courses, so it definitely opens up your mind, because it is not only like the lecturing part, but the hands-on part which is happening, be it quality, be it lab, be it analytics, be it clinical trials, operative services. It is all been there at the part of this course. And I am happy to announce that this is a credit-based, competency-based curriculum, and the credit goes into ABC. I hope the students knows what is ABC. It is not airway, breathing, and circulation, but it is the academic bank of credits. Nowadays, UGC and the NEP 2023 is coming up with a system that whatever you study, the credits are being generated and they are being uploaded, so that once you are putting up your CV, it is the credit which you have earned really defines the kind of research acumen or research experience or the experience which you hold. So the this once you do this course, it has got credits also which goes into the ABC, and there is an active learning from academic and industry experience. There are industry experts which are taking lectures directly into this, hands-on exercise and practicals, industry visit, industry internship, and project work is being given at the end of the session. This is 2 years course, which is semester based. And in the final semesters, you are being posted in an hospital where you are giving the project, where you really learn how to develop a protocol, how to conduct a study, how to interact with the patient, to not only develop the skills, but the communication skills also, which is very important nowadays in getting your patient. Because from my experience, I have seen that protocols are ready, but there is a lot of a difficulty in recruiting the patient. So that communication skill, how you need to convince your patient, how you need to take the informed consent, how you need to interact with the patient in the way that the patient really understands that how good this trial is for him, so that the ethical part is also taken care of. And once we are taking such patient into a clinical trial, we are progressing towards research also. So progression of research to ethics is something which is important and which is also being taken care of. And there is a separate module in this curriculum which teaches you that. Now, I think students would be like keen to know that what is the eligibility to join this 2 years credit system. So any health science graduate, be it MBBS, BDS, BMS, BUMH, BHMS, BPTH, they or even the nursing students like PNC nursing students, they can do this particular course. And an important thing is even the BSc life sciences students like Bicrop, doing BSc in life sciences in biochemistry, microbiology, botany, zoology, chemistry, physiology, pharmacology, and biotechnology, and also the pharmacy graduates. So the highlight, it's a postgraduate course which is being done, and the only ability is graduation. But let me tell you one thing, don't get biased by the word MSc. It is not MSc of Indian nature. It is MSc what is being considered in foreign countries. MSc in foreign countries is at par with the postgraduate degree. So this is something which is good for you people. I think good sessions have been taken. You have been sensitized. But if you really want to become a part of this industry, or if you are looking for a secure job option, if you have done this MSc curriculum, which has been conducted by MUHS, which is again to say a credit-based, competency-based learning system, which is approved by UGC, there is a lot of a chance of your better absorption into the pharmacology system and your better distribution to industry, which is looking for to take such people who are not only holding a degree, but they are skilled enough to handle operative works which an industry requires. I would just like my students to put up any queries if they want to know about this particular course. So I'll be really happy to, to guide them or to let them understand whatever I know about this particular course. I was heading this course as professor and head of the department, and recently I have been shifted from there to medical college. But let me tell you one thing: my heart and soul really remains with this particular course, because this is indeed good, quite interactive, where you get hands-on experience, one-to-one talking and learning from the experts. So with this briefing, I would request students to ask any questions if you want to ask about this particular course.
Thank you, sir. I'm very much thankful to Dr. Singh to concise talk on today's session conducted by Dr. Akshay Dwarkanath sir and Dr. Shailendra Singh sir. Yes. Thank you, sir. Any query from any participants?
Yeah, Dr. Weber, if you can really ask students if they want to know something more about
Yes.
this particular course. Any question in the chat box?
No, sir. In chat box, no question, but the session also live on YouTube. If any question, definitely we'll communicate you, sir.
Yeah, thank you so much for giving me this time once again. Thanks to Dr. Weber and Dr. Prashant sir for giving me this opportunity to remain associated with you people still. Thank you so much for this.
Yes, thank you, sir. Now I request Dr. Prashant Shugunde, presently working as an assistant professor at Department of Pharmaceutical Medicine at Maharashtra University of Health in Nasik. Dr. Shugunde will introduce today's session of MSc Pharmaceutical Medicine program. I request Shugunde sir, please proceed the session.
Yeah, thank you very much, sir, for your kind words. Students, as our Dr. Singh sir said, that this pharmaceutical medicine course is available at MUHS. I hope you might have, most of you might have hearing it first time. So let me just share some of the snapshots related to this information. This course is named as the Master of Science in Pharmaceutical Medicine, and this course has been run by Department of Pharmaceutical Medicine of the Maharashtra University of Health Sciences, Nashik, Maharashtra area. This course is located in Nashik. Though it is located in Nashik, we do have this course in such a way that we are taking it some of the sessions online. We do have a tie-ups with some of the government institutes where the students will be taken out and they will be given a hands-on experiences over there. So just to start with the why, what is this pharmaceutical medicine? I would like to just say that it is just a medical scientific discipline where we will learn about the discovery, development, evaluation, registration, monitoring, and medical aspects of marketing of medicine. So most of the time you might have heard of word drug, but this is what the difference that we are getting over here. We are learning about a pharmaceutical medicine. This course now a time the UGC has came out with the, rather than calling it as a course, it will be called up as a program. So this program has been started in MHS in 2007, and this is the first ever course approved by the UGC of the Maharashtra University of Health Sciences Nashik in the pharmaceutical medicine. No other, I hope a course has got such a UGC approval for the running a pharmaceutical medicine course, which is related to the preclinical as well as the clinical matters of the drug and medicines. I hope as Dr. Singh has elaborated out a detail, this course offers an interesting, challenging career as a professional in a drug product development. Most of the alumni have made a good career and they are pursuing a successful careers in academia as well as a industry in India and abroad. So this is just a short introduction about this course. If you can look out about the program outcomes at the end of this pharmaceutical medicine program, whoever be a learner will become a professional with in-depth knowledge of pharmaceutical medicine and its role in a healthcare, a professional with an aptitude and skills to conduct research and innovation in a drug discovery, development, and its use in a healthcare. We will also become an empathetic communicator who respects stakeholders, including colleagues, students, trainees, ethics committee regulators, industry, and society. This component gives a holistic approach as required by a UGC new curriculum implementation. Leader of a team and lifelong learner in his or her role as a researcher, teacher, entrepreneur, or industry expert, as well as an ethical practitioner in research, those who are from a health sciences background, socially and environmentally responsive to the problems at a local and global level. So we are not only making a student as a money-making machine, but we are also making him to be a socially responsible in whatever he or she is going to do. If you can see over here, we do have a 4 semesters. This program comprises 4 semesters, where is a first semester has a 5 courses with the total credits of 20. Then second semester has a 5 courses, again total credits with the 20. Third semester again have a 5 courses with total credits of 20. And the last semester deals with the project and electives. So let's see what are this semester 1 has. The semester 1 has a course 1 which tells about what is a MSc Pharmaceutical Medicine program, as well as it orients about the allopathic subjects pre, para, and clinical. In this course, we are sending the students to the civil hospitals or some of the hospitals, private hospitals, where they will learn about the different case studies and they will learn about how to take a patient, how to read a patient report, and how to take it or how to capture it into a electronic or a data form onto the forms and to find out the case studies. In a course 2, we are dealing with the general and clinical pharmacology, where the students will learn about whatever you have learned in a graduation related to the pharmacology, entire pharmacology, as well as some of the practicals. In course 3, drug discovery and pharmaceutical development, where we will learn about how a drug candidate from hit to lead proceeds and how its pharmaceutical development from the primary production to the secondary production into the market will lead. In course 4, the students will be oriented about the different analytical methods, what is the quality control process, and how a quality assurance is have importance in finalizing a medicine into the market. In a course 5, again we will deal with the preclinical studies, safety and toxicity testing. Whatever the sessions right now we had in the last couple of hours will be one of the just a snapshot. However, in this preclinical studies, we are actually dealing with the how a drug will be taken into the in vivo, in vitro, and ex vivo system, how it will be evaluated, and how a report will be written for these preclinical studies. If you can see for the semester 1, all these courses will deal with the most of the time a preclinical part. They have focused on a preclinical and the basic chapters. In semester 2, you can see that we have taken a course 6 as the development of medicines with the clinical trials. Then course 7 deals with the biostatistics and data management. Course 8 will deal with the pharmacovigilance. Course 9 will deal with the legal, ethical, and regulatory issues. Course 10 will deal with the pharmaceutical industry. Over here, you will see the shift from a preclinical to the clinical, where you will learn the basics related to the basic and the advanced things related to these clinical trials. What are the legal, ethical, and regulatory issues from getting it from a IND as well as a to NDA, that is a new drug application, as well as a post-marketing surveillance. In pharmacovigilance, you will be studying, you will be oriented about the all the adverse drug reaction classifications, how to do a causality assessment, how a preventability aspects will help in making a policy making, and the practical parts of it, where the actual entry into the Vigiflow will be taught. In semester 3, you will see economics of healthcare. Then in course 12, you will see medical and scientific writing and communications. In course 13, we will deal with the plant drug development and clinical phytopharmacology. In course 14, human genomics and biotechnology. In course 15, new drug delivery system and the other technologies. In economics of healthcare, you will understand that how a quality of health is important and how a cost of the drug is related to this quality of health of the society. In medical and scientific writing, the how to write a publications and how to effectively communicate to the regulators related to matters of the medicine approval. That will be taught into the course 12. In plant drug development, you will learn about how a drug from the plants will be extracted out, how they will be standardized as per the different WHO guidelines and the national and international guidelines, and how this will be applied into the humans by using a different steps of animal studies, in vitro studies, and the in vivo studies. In human genomics and biotechnology, again, basic as well as what are the therapies are available for the actual for the treatment of a different diseases related to the gene level will be taught in this structure. In new drug delivery system, what are the available different technologies are being applied right now for the formulations, and so as to optimize a drug will be taught into this course 15. So you will see that there is a shift in a finalization part of all medicines in the semester 3. In semester 4, you have to do an actual project on either it could be on a preclinical area or the clinical area. We are mostly emphasizing to do a projects on the clinical area because there is a lot of job opportunities right now into the market. The projects will be based on the rational use of drug utilization studies, pharmacoeconomic studies, then different projects like clinical trial studies, post-marketing surveillance studies, pharmacovigilance studies, therapeutic drug monitoring. Likewise, area will be chosen. For the electives, you can go for a different types of a publication is two types of electives needs to be chosen out, where you can either look for a publishing a paper, attending a university workshops, as well as the attending some of the NPTEL courses and demonstrating your leadership through organizing conferences, workshops at departmental level. So likewise, you will be trained how to actually manage the event relevant to our health sciences disciplines. So this is how all the program will run. Already, recently we have revised the MSc Pharmaceutical Medicine course, where we have taken inputs from the academics, not only academics, from the industry as well as from students and the alumni who have passed out. And they have, and based on that, and as per the UGC New Education Policy 2020, we have revised the curriculum. So this curriculum has ensured a quality curriculum where we are giving a outcome, competency, and credit-based, skill-based, where a practical work projects have been included. It makes students a job-oriented by attending industry internships. We are asking students to visit to the wards and OPD into the hospitals, into a rotations. We do have a tie-up with the civil hospital of the Nashik and some of the other hospitals, where the students will go and they will actually perform the task where they have to at least minimum for a first semester, they have to take up the 30 case studies. Principle of education technology, where we have applied to make it a student-centered. So this curriculum we are actually implementing from the last year, and we hope so this will make a holistic approach for training the students into the pharmaceutical medicine. So with this, I just end my session. Thank you very much, students. If you have any questions related to the course, you can ask. I'll just make you aware that how to reach to the curriculum content of this MUHS. You can just put a muhs.ac.in into the web browser and then go into the academic tab over here. I hope it is getting displayed to you all. Can you see a web browser?
No, sir.
Okay. I'll reshare once again. Okay, can you now see?
Yes, sir.
So in the web browser, you can just put a muhs.ac.in, and once these websites get open, you can go into the academics, where a courses and syllabus list will open, drop down list. Out of that, you have to choose a faculty as a medical and course type as a postgraduate. And on the second page, at the downside, you will get a syllabus of the MSc Pharmaceutical Medicine. So you have to just click onto the syllabus and it will show up your a detailed syllabus. Just go through the syllabus, you will find it interesting. What are the competencies that are being getting developed? What are the different opportunities? How credits have been allocated? What are the hours of learning? I have just forgot to mention over here that each course has been divided into a different types for the learning. One is a lecture, then self-learning, then tutorials, then practical, and finally experiential learning. This experiential learning will be provided by visiting to the collaborated institutes like National Center for Cell Sciences in Pune, National Institute for Research and Reproductive Health in Mumbai, KIM Hospital in Mumbai, National AIDS Research Institute in Pune. So there are multiple MOUs have been done through Maharashtra Institute of Health Sciences, Nashik, across the Maharashtra, where students will be deputed for the selective purpose of learning. So this is how this course will run for throughout 2 years, and this will also add a benefit of adding up credits into the academic bank of credit as per the new educational policy of the UGC 2020. So thank you very much, student, for patient listening for this session. If any questions you have, I'm open to solve any questions.
Thank you, sir. Providing the brief introduction of MSc Pharmaceutical Medicine postgraduate program, presently conducting at Maharashtra Institute of Health Science, Nashik campus. Thank you, sir. Now, on the behalf of MHS, that is Maharashtra Institute of Health Science, Nashik, I am very much thankful to today's speaker, Dr. Akshay Dwarkanath, sir, and Dr. Shailendra, sir, principal scientist from CSIR, Indian Toxicology Institute, Lucknow, Uttar Pradesh. I am also thankful to Dr. Jitendra Singh, sir, for providing the scope and potential of pharmaceutical medicine in pharmaceutical industry or in research. I am also thankful to MUHS ITT, Mr. Sachin Dinde and his team. Now I announce that today's session is over. In future, if you have any queries, regarding these topics or any further information, please communicate to DPM, that is the Department of Pharmaceutical Medicine, DPM small letters @MUHS small letters .ac.in. Once I'm again repeating, DPM, Department of Pharmaceutical Medicine, DPM small letter @MUHS.ac.in. Also circulate in chat box, departmental email ID. Thank you all of you. Now I announce today's session is over. Thank you. Have a good day.
Thank you very much, sir.
Thank you.
Thank you, sir.
So let me just share some snapshots related to this information. This course is made by the Pharmaceutical Science and Pharmaceutical Medicine, and this was by Department of Pharmaceutical Medicine. Housing vast list of success not Maharashtra, India. This course is located in the
याचं